Here we go. Okay. So we're recording. We're good. Let's start something new. How fun will this be? Okay, well, we're about to find out. Okay. Let's see if I can actually stretch this in here. Here we go. This is what I need. Okay. All right. So today, and actually, I'm not going to plug that in yet. I'll wait and see if anybody actually makes it to class. But today we're going to start talking about lipid biosynthesis. So note that we are jumping to chapter 21. We are skipping chapter 20 because we just don't have time to talk about plants, and we are going to be much more anthrocentric and deal with things that talk about things that deal primarily with humans. So, you know, we talked a lot about catabolism in that last section, how we tear stuff down. Now we're going to show how the body builds stuff up, and of course talk about the regulation of that. So the next three chapters are all about metabolism, where we are, kind of creating and building, and then looking at. I think we'll our third. I think 20, 21, 22, and 23. I think 23 is whole body regulation. So we'll kind of look at all that in there. My caveat is this is the dense stuff. It'll actually be kind of a relief when we get to the fourth section, because it is, it's like 5 chapters, but they're all shorter, and they're all, they're non-metabolism for the most part. But this is where, the semester every year kind of bogs down a little bit. So I'm trying to incorporate things a little bit differently, and maybe skim a few things and use some external resources. I found a few new supplements to post into the modules on Canvas, but I haven't gotten everything updated there yet. But I'll be working on that the rest of this week as well too. But we'll see if we can, make this a little more enjoyable. But this is the part where you're like, oh, here we go. Okay, so the biggest thing here in this chapter is looking at the biosynthesis of fatty acids. We'll talk about the eicosanoids a little bit as well too, because those are related to fatty acid production. And then we can use these compounds to make a variety of other bioactive molecules in the body. Then once we make our fatty acids, how do we assemble them into triacylglycerols? What about cholesterol? And our traffic and metabolism of cholesterol. So we're going to talk a lot about cholesterol in here, and then we'll look at the regulation and role of cholesterol. Why do we have like 3 bullet points about cholesterol? Because it's that important. You may remember some drug commercial a couple years ago where they talked about, it's like, where do you get your cholesterol from? Like your apple pie and your Aunt Vi or something like that, because there's both a dietary component and a genetic component. Cholesterol is important enough that that we need to consume it and make it de novo as well too. Okay. All right. Oh, awesome. Good. Okay. So let me project here. We're kind of doing a 2 for today because I got a couple sick people, so we're both Zoom and in person. So, and we just jumped into chapter 21, which maybe, there we go. Maybe. Yes. Come on. There we go. Awesome. Okay. So we'll be doing a lot of talking about generation of fatty acids and cholesterol as well too. Okay. So let's just briefly kind of talk about what lipids do in the body. They have several roles. Obviously energy storage. I'm storing a little too much energy now too. It's like, yay. We look at our membrane constituents, all of our phospholipids and so on in there. They can function as anchors for the membrane proteins, cofactors for enzymes, signaling molecules, pigments, detergents, transporters, antioxidants. If we go way back to chapter 10, I believe, it talks about a lot of these as well too. I always think about this too when you look at various birds. I don't know if anybody has bird feeders or stuff like that, but we'll see cardinals in our backyard as well too. And of course, the male cardinals are very bright red. That's because of the seeds they eat. If you compare them with finches, they have a different diet, and the compounds that they consume in there contribute to their yellow color. It's like, well, female cardinals eat the same thing, but they've got slightly different biochemistry, so they don't get that brilliant color in there as well. Anyway, that goes down a different track here. So we saw in the previous chapters that our catabolism of fatty acids is going to produce acetyl-CoA and give us reducing power, NADH and FADH2, and is going to take place in the mitochondria. So when we look at anabolism, we're going to start with acetyl-CoA and malonyl-CoA to build our fatty acids. We're going to require reducing power, which is going to come from NADPH. All right, so we got a different thing in there too. And this is going to take place in the cytosol, okay, or the chloroplast for plants. But we're ignoring plants because we don't like them. So we've got, basically completely different pathways that are going on here. Okay, so we want to keep them separated. That's why our anabolism occurs in the cytosol and our catabolism takes place in the mitochondria, because we're going opposite pathways. And it's kind of neat. This is a subtle difference here. We are producing NADH, but when we make our fatty acids, we're consuming NADPH. Okay, so we just get that reducing power from a slightly different molecule. Okay, so where is everything up here? Let's see. Yeah, it does show up on there, doesn't it? And let me see if I can minimize that a little bit there. There we go. Now we can see slides a little bit better. And this is just kind of showing where everything is and stuff in here. This is like just a good general reference, a good biological reference in here. Okay. Oops, come on. There we go. Okay. So let's look at our overview. Just like when we break down our long chain fatty acid, when we cleave off 2 carbon units at a time, our fatty acids are going to be built up in a very similar way. We're going to process 1 acetate unit at a time. So we're going to add 2 carbons at a time. Okay. In this case, instead of acetyl-CoA, our acetate is coming from an activated malonate. So this will be our malonyl-CoA. Okay. So you can see our malonyl is our 3-carbon subunit. You might remember if we had an odd-numbered fatty acid, we'd be left with malonyl-CoA. So what's going to happen as we build this up, we're going to add 2 of these carbons to our growing fatty acid chain. All right. Basically, we're going to reduce that carbonyl carbon to a methylene carbon. Okay. We require NADPH. Where does that come from? It's going to come from the pentose phosphate pathway and the malic enzyme a little bit as well too. So basically, as malic acid gets converted into pyruvate with CO2 left over, we get our NADPH. We also have our lovely pentose phosphate pathway in there. And it's kind of neat. You can see like our fat cells have a different way of generating NADPH as opposed to our liver cells. Because we're looking at glucose 6-phosphate being converted to ribulose 6-phosphate. Okay.
You're saying the carbons get attached to what?
To
the starting.
So we'll get to that. We'll get to that. Yeah. But we're basically going to take all these malonyl-CoAs and then take 2 of those carbon units and just keep adding them up to a growing lipid chain. Yeah. And then we'll have to, just like how we saw breaking them down, we had like a 4-step process to break those down. We're going to see the processes that would go to add them and then oxidize or reduce them as needed. Okay. Okay, so we can see here, malate getting converted to pyruvate in our adipocytes, and glucose 6-phosphate to ribulose 6-phosphate in the pentose phosphate pathway. And we didn't go into that in any huge great detail, but that's one of our fates of glucose. And you can see we get a couple of NADPHs over there. So as we go into our pentose phosphate pathway, we get NADPH. We can use that energy to make our fatty acids. Okay, and again, this is here we have a carbohydrate. We've brought our glucose into the cell. We phosphorylated it, and instead of going through glycolysis, we shunt it over to the pentose phosphate pathway. Okay, so how do we get our malonyl-CoA? We're going to combine acetyl-CoA and bicarbonate. All right, so and again, we're going to have let's see. It's about the right time. Usually, our semester starts a little earlier, so I can talk about the NCAA tourney and the ACC acetyl-CoA carboxylase in there, right? Okay. So our acetyl-CoA carboxylase is, again, another multifunctional enzyme. It's got 3 subunits. We've got biotin in there. So we know, oh, biotin, that helps us carry single carbon units. So maybe that's where our bicarbonate is going to come from. In this case, it's actually carbon dioxide, but along the same lines there too. For us in all animals, all the subunits are on a single polypeptide chain. Okay, so our bicarbonate is our source of carbon dioxide for the biotin that's going to interact it with the acetyl-CoA to make our malonyl-CoA. Okay, so let's talk about our reactions. Let me close the door to get less external noise. All right. So, multifunctional enzyme here. We have this lovely biotin carrier protein with this biotin arm that's going to hold on to our carbon dioxide that's coming from bicarbonate up here. Okay, so we're essentially going to spin some ATP to attach this on here to our biotin carboxylase. So this is very similar to our pyruvate carboxylase that we saw. And it's also similar to propionyl CoA carboxylase in here. So we bind this CO2 to the biotin, and then you can see that our biotin is going to be able to swing over to that other part of the enzyme for the next step. Okay. So as our biotin swings over here to the trans-carboxylase portion of the enzyme, we can have our acetyl-CoA come in and bind, and then the trans-carboxylase will catalyze the carbon dioxide attachment to the acetyl-CoA, to our carbon here, to generate malonyl-CoA. Okay. So our trans-carboxylase, we're transferring our carboxylic acid group over there. Okay. All right. Once this is complete, our malonyl-CoA will then fall off the enzyme, leave the active site, and our biotin is free to swing back over and pick up another carbon dioxide. Okay. All right. So this is kind of our two-step process to generating our malonyl-CoA. Okay. So once we have our malonyl-CoA, we can move on into a fatty acid synthase. All right. And this is going to catalyze a four-step sequence that elongates the fatty acid chain. So just like we had a four-step sequence to cleave off, and it reverses that.
I can't hear you anymore.
Oh, okay.
Oh, there we go.
Is that better?
Yes.
Okay, good. I think I probably just had something covered there. Okay. Okay, so yeah, but definitely check out this figure in here. This will be a good figure in there. I also like I say, Lippincott's Illustrated Review has a good illustration of fatty acid synthesis as well too. Okay, so we're going to use, besides our NADPH as the electron donor, we're going to have a couple sulfhydryl groups in there. We basically have a couple different versions of fatty acid synthase as well too. We're going to be primarily concerned with isoform 1, FAS1, because that's, we're in vertebrae. There's a different fatty acid synthase in plants. Okay. All right. The big differences between these two is, the animal one is a single polypeptide chain. There are multiple peptides in plants. Our animals, we only make palmitic acid. Plants can make a lot more variety of fatty acids though. So we only make palmitic. Once we make palmitic acid, we can modify that later on, but fatty acid synthase 1 only makes palmitic acid. Okay. And then our initial carbons in here are from the acetyl-CoA that's used to prime the reaction. Okay, we'll see that here in a minute. Okay, all right. So here's our fatty acid synthase 1. You can see we have almost kind of a dimer looking shape in here with a couple of these different active sites in here. All right. So more on these coming up in a minute. This is just a lovely three-dimensional structure of it, kind of looking down on top and looking at it from the side. So what are all these different active sites? Well, let's talk about them. Okay. So our overall goal is we're going to take a 2-carbon unit from malonyl-CoA and attach it to a growing chain of fatty acid, and then we need to reduce it. Okay, so here are 4 steps. First thing we start with is a condensation to get them connected. We're going to reduce our carbonyl group to a hydroxyl group. We're going to dehydrate the alcohol to a transalkene, and then we're going to reduce the alkene to an alkane. Okay, so we basically have to get rid of those oxygens that we're bringing in. Our oxygens help make this, an activated compound that make the reaction more likely to occur. Once we've attached the acetyl group in there, we need to get rid of that oxygen and replace it with hydrogens so that we can have this growing carbohydrate chain. Okay, all right. And of course, our chain is attached from this thioester linkage from one of those sulfhydryl groups that's in there. Okay, so our preparation, we get malonyl-CoA and our acetyl-CoA, or if we have an ongoing chain in there, but assuming we're starting from the beginning, acetyl-CoA, they are both bound to fatty acid synthase 1, and they lose the coenzyme A. Okay, so we're binding them via a sulfhydryl group in there, that thioester in there on the fatty acid synthesis, and this will activate the acyl group. Okay, so once we have them bound, we can do step 1. We have this condensation reaction that'll attach 2 carbons from the acetyl group to 2 carbons from the malonyl group, and will release carbon dioxide. Okay, so you think as we're making fatty acids, take a big exhale, relax. Well, what do you do after a big meal? You have a nice big relaxing and breathe out carbon dioxide. I don't know if that's helpful or not, but we'll have this beta keto intermediate. Okay. Do I have a picture of that? Well, it's probably coming here. Okay. So we're essentially making this acetoacetyl ACP. This is a Claisen condensation. Yay, organic chemistry, right? Okay. We have this particular enzymatic portion of our fatty acid synthase of this beta keto acyl ACP synthase. All right. So this would be our, I believe, our KS. So we go back up to our picture here. Our KS region down here in the middle, that's where we're starting from, okay? For keto acyl ACP synthase, okay? And then we couple this to decarboxylation of the malonyl-CoA to make it energetically favorable, okay? So here's probably our better payoff picture. Here we have our 2 thioesters that are formed, and then our enzyme catalyzes this reaction where we end up seeing our carboxylic acid, the lone pair here, drops down onto his so that we can make our carbon dioxide a leaving group, and our electron bond, our bonding pair here can shift from this carbon to the one on the acetyl-CoA. Okay, and then this will release from this sulfhydryl group, and we now have our beta ketoacyl ACP that's here. Okay. So our next step, we're going to see this reduction that comes in. Okay, so we're going to reduce this carbonyl group, our beta carbonyl group, to a secondary alcohol. Okay, so our first reduction, NADPH, is reducing that beta keto to an alcohol. Okay, get that in there, and this is catalyzed by the Kr portion of our enzyme. So if I go back up to that picture again. All right, so we've got our Kr. Where is that shown up here? So that's kind of up here in this little section, which looks like it's really far away, but it's not all that far away. But again, you can see all these little regions in here are involved in this chain of catalysis that's occurring. Okay, so step 2, we reduce that carbonyl to an alcohol. Okay. Once we do that, we have an alcohol group. Well, that's a good leaving group. We can toss it out. We're going to go on a temperance movement. We're going to get rid of the alcohol. So we're going to dehydrate that hydroxyl group. So we're going to take the hydroxyl group from carbon 2 and a hydrogen from the neighboring group and eliminate that to make a transalkene. Okay. And this is done by beta hydroxyacyl ACP dehydratase, or our DH subgroup. You don't really need to know all that. You just need to know the condensation, reduction, dehydration, reduction. Okay, so our last step, we're going to reduce that transalkene to make a saturated alkane. Okay, and this is done by the ER portion in there. Okay, so here we are with our lovely little alcohol group. We are going to remove the alcohol, okay, along with one of the hydrogens over here on carbon 2, okay, and make this transalkene, and then we reduce the alkene to make our alkane. And now we're ready to do this again to our growing chain. Okay. Okay. We'll do the same thing where we'll see, our this will occur here. All right, we can bring in another malonyl CoA. Okay. Do the same type of deal where we will release this, add another. So you can see that basically we're adding this one down in here, we're adding this one down in here, so on and so forth until we have a long chain of 16 carbons. So basically do this 7 times. Yeah, so we have eight additions in there. So we end up making our palmitic acid. So this is our product of fatty acid synthase 1. Okay. All right. You just kind of keep doing that, keep going with that transfer. Okay. Okay, so hopefully that makes reasonable sense. Okay. All right. Once we have that, we're going to use an acyl carrier protein as a shuttle for the fatty acid synthesis. Okay. So our acyl carrier protein carries acyl groups. That's what it does. So it's got a prosthetic group of this phosphopanthine in here. And it's, again, another one of these nice long flexible arms so that we can move stuff around. And it's going to deliver the acetate in the first step or malonate in all the next steps to the fatty acid synthase. And it shuttles the growing chain from that one active site to the other. So if you note, like, wait a minute, okay, here's our malonyl CoA that binds here, okay? But when we make our product is on that same site. So our ACP is going to help shuttle that growing chain from this site to the other site. So that's why we have our 2 sulfhydryls. So we make our bigger chain here, but then we move it over to this sulfur. And then we add another one onto here and then shuffle that over to get this. And then we add another one and so on and so forth. Okay. So our acyl carrier protein is just kind of shuttling our growing fatty acid chain back and forth between the,
Does this all happen in the mitochondria?
this is all in the cytosol. Okay, all in the cytosol here. Okay. So the thiols in there, those sulfhydryl groups, they need to be charged with the correct acyl groups before the condensation can begin. Okay. Once they have the right ones on there, they get activated and this can occur. All right. So that MAP region of fatty acid synthase
I can't hear you.
Oh, goodness. All right. I think maybe I'm covering up my microphone. So I'm just going to stop touching. Can you hear me now? Uh-oh.
Better.
Okay.
Yes, I can hear you now.
Okay. Yes. I need to stop touching. I apparently where my hands fall, it covers up the microphone. I just need to, break down and get a headset. All right. Okay. So again, if we look at this in some detail, we have all these different regions here. And essentially what's happening is our acyl carrier protein portion of this is just kind of moving stuff back and forth between KS and KR. Okay. I'm not going to have you guys go into excruciating detail about that. The main things I want you to know is it's a 4-step process for our fatty acid synthase. Know what we start with, acetyl-CoA and malonyl-CoA, and then the 4-step process in there, that condensation, reduction, dehydration, and second reduction. And then ultimately we end up making palmitate. Okay. And that our acyl carrier protein is involved in putting our growing fatty acid chain where it needs to be. Okay. All right. Sweet. Yeah. There we go. Growing chain. Yeah. So all this kind of bends around here. And you can see with this kind of this 3-dimensional shape that you do need that acyl carrier protein to connect that KS region that was on the bottom with that KR region that was kind of on the top in there. Our acyl protein shuttles them back and forth. Acyl carrier protein shuttles them back and forth. Okay. So here we start, just kind of visualizing this in space. Our condensation reaction starts here. We charge our acetyl-CoA with our KS region and our acyl carrier protein holding onto our malonyl-CoA. We attach this on here. we move this up to KR and so on and so forth. Then our KR can reduce it. And then we can slide it around to the DH region for our dehydration, and then our elongation reaction, reduction, all our good stuff in there, so on and so forth. So again, our acyl carrier protein is involved in moving all this, and then it slides it back to our KS for the next round, adding another acetyl group. Okay, so it's actually kind of neat. It's I'm trying to think. Seems like there was a childhood toy, and I just can't remember the name of it, where you'd have something that would some chain that would fall in different positions on some toy or something like that. But and you're just kind of cycling through here to put our various components where they need to be to allow the enzymes to do their thing for these 4 steps. Okay. Okay. That's what I really want there. Okay. So condensation, reduction, dehydration, reduction, and then chain transfer, starting, setting it up for the next round. Okay.
And so we don't need to know those.
You do not need to know the things in red. No. Now, if I ask you a question like that, it'll be in the take-home section in there. But yeah. But condensation, reduction, dehydration, reduction. That's where we are. Okay. So let's talk about the stoichiometry a little bit. We need 7 acetyl-CoAs. We make 7 malonyl-CoAs, and all these are going to use ATP. Okay, so yes, yeah, yeah. It's just that carboxylic acid on the end. A lot of places will write palmitate because it's a little bit shorter than palmitic acid, but they're the same. 16 carbon saturated fatty acid. Okay, so we take our 7 acetyl-CoAs to make our 7 malonyl-CoAs, consuming 7 ATPs and 7 carbon dioxides. So we have 7 cycles of condensation. So we need 1 more acetyl-CoA plus our 7 malonyl-CoAs that we've made, plus a whole bunch of NADPHs and some hydrogens here to give us palmitate. Okay, and you'll note that we're consuming 7 carbon dioxides. We also produce 7 carbon dioxides, so we're kind of recycling that in there, and we make some water. Okay, all right. So here's the neat thing. Our acetyl-CoA is made in the mitochondria, right? But fatty acids are made in the cytosol, so we have to transport acetyl-CoA from the cytosol, from the mitochondria into the cytosol, and that is expensive. It costs us 2 ATPs. Why is it 2 ATPs? Well, remember, mitochondria has 2 membranes, so 1 ATP to get across the inner membrane, and another ATP to get across the outer membrane. So essentially, our fatty acid synthesis is 3 ATPs per acetyl group, right? So 2 for the transport and 1 for the actual activation.
When you were breaking down the fatty acids, you had those transporters. Are you gonna have those are you gonna use the same transporters to get it across the membrane, or are they
well, not really, because they are different because we're bringing in large, anywhere from small, medium to large fatty acids in there. So there's 3 different ways they can get in. Like the small ones can just freely diffuse across, but because we're exporting acetyl-CoA, that coenzyme A is not attached as the fatty acids that come across usually. So we have a different method for getting them out. And we're just bringing out that acetyl-CoA 2-carbon unit in there, okay? And this is essentially how this works. We can look at our aspartate, wrong transporter there. We got our pyruvate transporter and our citrate transporter in here. So here we are inside our mitochondria. I always look at this backwards because I'm geared to thinking that the right-hand side is the inside and the left-hand side is the outside, but no, it's this section. This is our mitochondria here. So we make our acetyl-CoA, okay? If we look at our citrate synthase, right, from our citric acid cycle, okay, we make citrate. We can have our citrate transporter send it outside, okay? And then our citrate can be acted on by citrate lyase to make oxaloacetate, but ATP utilizes this to make, one of our side products is the acetyl-CoA. So in essence, we're using citrate to transport our acetyl-CoA outside. So we're not actually physically taking acetyl-CoA from one side to the other. We're taking a metabolite of acetyl-CoA, citrate, transporting that out, and then regenerating it back into acetyl-CoA. We make oxaloacetate, which can be converted to malate. So malate can go in that way, or we can convert it to pyruvate. Send pyruvate in there, and then we can use ATP to convert pyruvate to oxaloacetate. So this is where our ATP expenditures are coming from to export this out of here, because we need to include the ATP we're spending for our regeneration. Okay. All right. Whew. Good times, right? It's a lot. Okay. Yeah, it's funny because tracking this down is kind of like we were doing some changes to the program. We're trying to calculate all the different credit hours and where they come from, and I keep getting different numbers. I'm like, I swear, whatever our old paradigm was on this particular area, somewhere we have an extra 2 hours. I don't know where those came from. Maybe we're counting a class twice. Maybe there was another lab in there. Maybe we just require 2 independent researches. I don't know, but it's like there's got to be an error because I cannot figure that out. But same way here it's all about bookkeeping. Where does all this come from? Where are we spending our ATPs? Where are we making our ATPs? All right. Okay. So again, our fatty acid synthesis is very tightly regulated, and it's going to be our acetyl-CoA carboxylase enzyme that catalyzes a rate limiting step. So that's going to be our target of regulation in here. Okay. So we got a couple things that happen. ACC experiences feedback inhibition from palmitoyl-CoA. So once we have our palmitic acid, while it's still attached to coenzyme A, that can bind to ACC and inhibits its interaction. So that's feedback regulation, feedback inhibition there. On the other hand, if we have lots of citrate, so if we're transporting lots of citrate out of the mitochondria, we end up with activation of ACC. So we have basically, in essence, we have a large amount of acetyl-CoA, so we can utilize that to make our fatty acids, okay? And what it says here, our citrate signals that we have excess energy, so we can convert that to fat for storage later, okay?
So citrate is the rate limiting?
So acetyl-CoA, the ACC is the rate limiting enzyme, and then citrate activates that. Okay, so our mitochondrial acetyl-CoA gets converted to citrate, which goes to the cytosol, where it can be converted back into acetyl-CoA. But if you have a buildup of citrate, it'll bind to ACC and say, "Go faster!" So that's what we got going on there. What else do we see here? Any hormones that indicate a need for energy will inhibit ACC. So glucagon says, hey, body's in a low energy state, I'm hungry, I need energy, something like that. Stop making fatty acids and start producing energy, okay? On the other hand, epinephrine, fight or flight, we're not gonna store this energy, we're gonna use this energy right now. And these act by causing a phosphorylation, which inactivates the ACC enzyme. And then when it's dephosphorylated, it will activate again. Okay. So our citrate is feed-forward activation, palmitoyl-CoA is feedback inhibition, and then our hormones can bind for a phosphorylation cascade. I guess they trigger a phosphorylation cascade. Kinases will actually bind it there. Okay. So that phosphorylation, ACC can also be regulated by covalent modification. Okay, so essentially when we phosphorylate it, the ACC, it reduces its sensitivity to citrate. Okay, so we can't bind citrate as well. All right. The neat thing about ACC is it is active as a bunch of dephosphorylated monomers. Okay, when it's dephosphorylated, that ACC can polymerize into these longer inactive filaments. All right, and the phosphorylation reverses the polymerization. Okay. Okay. All right, so that we essentially see these lovely little filaments here. So if we start hooking them all together, we deactivate it. If we dephosphorylate it, they unhook and become more active. All right? And they can be activated by citrate as well too. There are also other ways we can regulate fatty acid synthase as well too. We can look at gene expression. Okay, so various fatty acids and the eicosanoids, which are derived from our palmitic acid here, can bind to transcription factors. In this case, a specific class called peroxisome proliferator activator receptors, or PPARs, because that's a lot more fun to say and shorter. And those can go to the nucleus and activate the expression of the genes for fatty acid synthesis. Okay, so if you have a lot of fatty acids circulating, you're going to activate your fatty acid synthase. We will also see a form of reciprocal regulation. The malonyl-CoA will inhibit fatty acid import into the mitochondria. Okay, so if we have large quantities of malonyl-CoA, we're going to say, hey, fatty acids stick around out here. We're making more of you. We can send you to the adipocytes or something like that. We don't need to burn these fatty acids. We're making fatty acids, so we will store these. Okay, so this reciprocal regulation is just a way to avoid a futile cycle, so that we're not sending fatty acids to the mitochondria to burn them for energy, and then bringing acetyl-CoA back outside to build up fatty acids in here. We don't want both processes going at the same time. In a well-fed state, we're going to be building fatty acids and storing them for later. In a state where we need energy, we're not going to be building them. We're going to be tearing them down, sending them to the mitochondria to use that energy. Alrighty. Okay, so let me check where we are. Okay, good. Oh, wow. Yeah, we're fine. We're fine on time. This is good. So, ultimately, our fatty acid synthase 1 makes palmitate. Okay, so once we have our palmitate, what can we do with it? Well, we can use it as it is, or we can modify it. Okay, so we can lengthen it to make longer chain fatty acids. We send it to the endoplasmic reticulum or the mitochondria and create longer fatty acids in here. And again, we typically add in 2 carbon units in here. So stearic acid or stearate is our most common product. Hey, we'll just add another one on there too. It's like 16 is good, but 18 is better. We can also start introducing, do some desaturation in here, so that we can make palmitoleate. Basically, we introduce a double bond between carbons 9 and 10. Same thing, we can do this with stearic acid. We can make oleic acid and get an 18-1 fatty acid in there. So this is a fatty acyl-CoA desaturase. Okay. Basically, there's a class of these, fatty acid desaturases. And again, just like with building our fatty acids, our fatty acid synthase, we need NADPH. And we're going to use some various cytochromes for our reductive power in here. Okay. This is kind of neat because in this case, our fatty acyl desaturase is a delta 9 desaturase. That basically means it's going to be reducing specifically the carbons, the bond between carbons 9 and 10. Okay, if we wanted our double bond someplace else in there, we would need a different desaturase. So the way that this desaturase works, it's going to say, I'm going to reduce this particular bond. All right. Essentially, this is the way it works. We can see our NADPH transfers electrons to our cytochrome, to our cytochromes, which transfer them to other cytochromes, which provides the energy for our fatty acyl desaturases to make that monounsaturated fatty acid. Okay. All right, and we could go into huge detail about this, but that would be its own specific course, so we're not going to do that. All right. Neat fun fact time. We have delta 4, delta 5, delta 6, and delta 9 desaturases, but we can't desaturate anything beyond carbon number 9. Okay. So plants can make, oh, sorry. Yeah. Gotta pull my hand back there. All right. Let me put like a little electric shocker there. But plants can produce things beyond there. So they can make linoleic acid. Think of like olive oil for cooking in, and alpha-lin. So they have like delta 12 and delta 15 desaturases. These are in effect essential fatty acids to humans because we need them to make other eicosanoids, but we cannot make these fatty acids ourselves. We have to get them through our diet. So just like we have essential amino acids, we have essential fatty acids as well too. Our various polyunsaturated fatty acids, or PUFAs, help us regulate the membrane fluidity as well too. So if you think back to our membrane, we can thicken it with cholesterol. If we start adding in a bunch of polyunsaturated fatty acids as our side chains for our glycophospholipids, then we see we have a more fluid state. If we have more saturated fatty acids, it becomes a firmer, less fluid state.
So we get that from plants?
Yes, yeah. Okay. Our plant desaturases act on these on fatty acids on a phospholipid. Okay. So essentially, we need to have a phospholipid there, and then the enzyme can bind that phospholipid and add in another double bond or 2 or 3 for like arachidonic acid and all that good stuff in there. Okay. All right. And that's all we're going to say. Yeah, then we can then hydrolyze this and release this fatty acid from the phospholipid backbone, but this is just what's necessary for plants to do their thing. Okay. What do we have here? Pyruvate, oxaloacetate. Okay. Okay. Yeah. Not sure what I want to say about this. Do I want to say anything about this? Come back here. No. Okay. Nope, that's woo. What did I do there? Okay, that's exactly what I wanted to do. And no, I don't want to turn that on. So okay. Oh crap! How do I get rid of that? That's a good question. Oops. Well, anyway, no subtitles. It's weird. I should just be able to close it without it. Anyway, okay. Yeah, so we make all this lovely stuff in here. Okay. So let's talk about, this is really weird. Let's talk about our eicosanoids briefly. Okay, and this will probably be where we stop for today. Our eicosanoids are a class of hormones that are made from arachidonic acid, or arachidonate, okay, which is a 20 or 24 carbon fatty acid in here. Our eicosanoid hormones include the prostaglandins, our leukotrienes, our thromboxanes. So basically, you're looking at any type of immune response or inflammation or stuff like that. You got to have this in here. So basically, we start with arachidonic acid, and we incorporate that into the phospholipids and membranes, and then we can use that to make our various eicosanoids. So various stimuli, whether it's a hormone or injury or whatever, will activate this phospholipase A2, because our arachidonic acid is going to be on the second carbon of the triacylglycerol. It will be activated, attack that C2 fatty acid, and release the arachidonic acid. Okay. So with our arachidonic acid that's up here, that's interesting. I wonder what you guys are seeing there. Anyway, we can have this, in this case, prostaglandin H2 synthase is a cyclooxygenase / peroxidase that functions in, that is positioned in the endoplasmic reticulum. Again, we go through a bunch of steps, but we can see here is our arachidonic acid. That's weird that shows up there, but it doesn't show up that way from, all right, well, we're covering something up there, but basically this is, just our phospholipid up here. And this is our arachidonic acid. We just draw it this way because of how we're gonna do the chemistry on it. Normally, if we were just talking arachidonic acid, we'd draw that as a nice long straight line. But we bend it around this way because that's what our cyclooxygenase is gonna do. And then we're gonna add our oxygen across here, and we're gonna form this ring structure in here. So we have this nice little bridge as well too. And then we can see that we start activating this too with some oxidation reaction in here. And we're going to end up with our PGH2 molecule, which then we can make other prostaglandins, or we can go into a different pathway and make thromboxanes from this. Okay, so our PGH2 synthase has two isoforms. There's COX-1 and COX-2. And again, our COX is coming from cyclooxygenase. Okay, so COX-1 synthesizes our prostaglandins that regulate mucus release in gastric mucin secretion. So you got to have your mucose. Must have stomach slime to absorb food. There we go. That's what I'm trying to say there too. So when you eat, you got to have that mucin to kind of lubricate the your food so that you can break it down and absorb it and bring it all in. So COX-1 regulates gastric mucin secretion. COX-2 catalyzes the synthesis of prostaglandins that are involved in pain, inflammation, and fever. So our various NSAIDs, aspirin, ibuprofen, acetaminophen, all inhibit COX-2. Okay, some of them are a little, more promiscuous. If we think of aspirin, anybody take bufferin, buffered aspirin, so that it won't irritate your stomach. A lot of these drugs, they can irritate your stomach. Why do they irritate your stomach? Because they also bind to COX-1 and inhibit that gastric mucin secretion. So you end up with stomach issues, and ultimately, if you take way too much, you can end up with ulcers in there because you're disrupting that mucin production, which is important for maintaining the health of the gut. All right, so our NSAIDs inhibit our cyclooxygenase activity. We have a nice, fun discussion in here, which is now in relative history. Aspirin is an irreversible inhibitor. It binds to it's in effect a suicide substrate. It essentially blocks the active site in both COX-1 and COX-2, and essentially removes those COX enzymes from the body, and you have to make new ones. On the other hand, ibuprofen and naproxen are both competitive inhibitors. They'll bind, but eventually they'll fall off. So they typically tend to be tolerated a little bit better than aspirin, at least as far as stomach upset. So if you go way back into history, people would say, oh, I'm in pain, let me chew on some willow bark. Why willow bark? Because willow bark was relatively high in salicylic acid, which is the precursor to making aspirin, acetyl salicylate, right? So willow bark tea would be good for treating pain. But again, it's not as concentrated as pure aspirin. Pure aspirin, I suppose if you gnawed down a whole willow tree, you'd probably get an upset stomach from all the salicylic acid.
Do you know why they use aspirin as a blood thinner?
I used to, but I've forgotten. I know. What does it do there? I'm not sure. I know it binds to more than just these inhibitors, but yeah. I'll have to look that up and get back to you on that. Okay. Oh, are we skipping our history lesson? No, we can't skip our history lesson. So, updated slides in here. So there was, I guess it must be ancient history now. I mean, if it happened in my lifetime, there was a new miracle drug, another anti-inflammatory drug that was specifically made to target only the COX-2 enzyme. So it'd be no stomach upset, only the enzyme involved in pain and inflammation responses, right? And what was the name of this drug? I'll have to look it up, but no, it used to be on the slides in here too. But it was great. It was awesome. People could tolerate it. You didn't have to worry about that. Is just attack the inflammation, and then people started dying of heart attacks from it. And it was just an unforeseen side effect, and it had a relatively high and lethal side effect. I mean, it's not like everybody who took it, but a significant number of people, and it was taken off the market and given black box labels by the FDA and essentially removed. I mean, it's gone from the market. It may actually be making a comeback on a different thing in there too, but I can't remember what it was. But if you look up COX-2 inhibitor that was pulled off the market, you'll find it in there. So yeah, everybody in the pharmacy industry was like so excited. It's like, yeah, we've got this new awesome drug that's going to make a lot of money, and then it started killing people. So Okay. If we look at our leukotrienes, okay, they also are made from arachidonic acid. Okay. So here we have, again, some different enzymes, whether it's lipoxygenases in here or our epoxygenases. We're going to make some epoxies in there. We will basically have this whole series of enzymes that can, again, it all starts from arachidonic acid. Okay.
Oh, they still have Celebrex. I didn't know that was a tox too.
Yeah.
They said that's the only one they left on the market.
Yes. Yeah. So actually, let me pause here with this because we're about halfway through, and this is a good stopping point. I've forgotten so many of the.
Extra and Biox.
Maybe it was.
Off the market in 2004,
I think it must have been Vioxx. That was the one that I remember hearing about. So, not full screen. I want to undo that. No. Okay. There we go. Okay, let's shrink this down for a second here. Okay. Oh yeah, I can't bring it in.
That was a Pfizer drug, Extra.
Okay, it was Vioxx. Yep. But I remember seeing. Maybe there's another name for it as well too, depending on there. But yeah, they were. They they had a. It's actually kind of neat here if we take a look at. Maybe I'll post this. This is a nice little health thing here.
And there's a warning on Celebrex, but they left that one on the market.
Yes.
A lot of people use that.
A lot of people use it. Yeah. So I think there were some dosing changes that were made to make it available, and they only use it for a particular type of pain, I think, and yeah, but not cool. But I'll post this as kind of like a little aside in there. Okay, no, thank you. Get through the ads, but learning from the COX-2 inhibitors there, it's actually really kind of neat. When my wife was in pharmacy school, they had to read a book about the FDA and how the FDA works, and for being such a small agency, it generally does a pretty good job, but it relies on information. They basically make the pharmaceutical industry pay for the testing. Stuff can sometimes sneak through, but for what it is, the FDA generally does a pretty good job in here. But this is just a short little article that kind of talks about, well, maybe not that short. I think it's short, but it depends on how much. But it talks about, what people learned from this, because essentially, Vioxx and Bextra were killing people. Yeah.
Did they just did they not have enough clinical trials, or they didn't have
Probably.
clinical trials?
Yeah, it was from and you whenever you have these big clinical trials, you have everybody has so many different health reactions. Like some people are going to die during a clinical trial, pretty much no matter what. When you're talking about like thousands of people, so why did they die? And so there probably wasn't enough power. And then of course, any company that's conducting a clinical trial doesn't want to, look at that data too carefully. But then once this part starts being prescribed to tens of thousands of people, and then you start comparing mortality rates, and you see that the risk of heart attack and stroke are doubled in people who have similar etiologies, and the main difference is whether they're taking this drug or not. That's when you kind of revisit that data and kind of look at that, and having this longitudinal data, and you can say, whoa, okay, we got to pull this drug back off.
I've noticed doctors don't pay attention to black box labels either, and they don't ask any questions or anything. But like, I was prescribed Ozempic for weight loss, and I have a history of thyroid cancer in my family. And there's a black box warning right on the front of the box, and it says, may cause thyroid cancer.
Yeah.
And I don't even understand why those are even on the market.
So, and I can help with this as well a little bit. So most doctors don't know crap about pharmacokinetics. It has changed over the years. It's funny, but what I've seen happen in large hospitals is you essentially have a care team now, and the pharmacists and the pharmacologists are in
They're very active.
Yeah, they need to be hands-on to kind of help out with all that, and make sure that the medication is appropriate and that there aren't any interactions and all that good stuff in there as well too. So having a good pharmacist, which is why I was talking in my previous class, the senior seminar, about people who are interested in pharmacy school. If you go to pharmacy school, almost no one wants to do it, but doing a clinical residency, which is a couple years at a very low salary because it's a residency, but it will make you all pretty much immune to the downturn that pharmacy is experiencing now. But yeah, it's probably a worthwhile investment. I know of multiple big job openings in Winston for clinical pharmacists because there just aren't that many people who do a residency. Meanwhile, there's always jobs for pharmacists as well too, but there's not necessarily the hours for them in retail. Yeah, you definitely have to have, most physicians, unless it's a very common drug, they just don't know that much about the drug. And this is it's an advantage to having a large amount of knowledge about biochemistry and pharmaceuticals and whatnot, because that really helps you advocate for yourself. And I'm the type of person, if I get a prescription, I'm going to look it up and try and figure it out and all sorts of stuff there too. I always like to tell the story of, like, I have an allergy to penicillin, although maybe I do, maybe I don't. I had an allergy like 30 some years ago, I think. So I haven't had any of the psyllin family in a long time, and it wasn't bad. But I did have one antibiotic that I was prescribed one time that, if you read into it, there are some weird mental effects that come into play in like a less than 1% of the people who take it. Winner. So now whenever I go to update my doctor, it's like, I was allergic to penicillin and levaquinone, or what I think that's what it was. And I'm not really allergic to it, but I do not tolerate it. It's like I get one of the crazy weird side effects from that drug, which is funny because I look at things like I've had what's the not morphine, the low level painkiller. It's like in Tylenol 3, I forget, but it's some drug that's supposed to be a painkiller. And I remember a bunch of my friends in high school were like, "Oh, you got prescribed that? That's so awesome! You're gonna get so high," and nothing. It was like taking regular Tylenol.
Well, that was the same with you with alcohol too, though, right?
Yeah. So
like opiate is not gonna give you anything, really.
Well, no, I did, because I did later on have morphine, and and that was. The physicians at Wake prescribed the exact right amount for me, because I've seen people in morphine hazes, and I'm like, I don't want to be out of it like that. But I didn't feel the pain that I was having, and I was still clear-minded. I was like, that's a good job, But that's helpful. That's useful. I remember when my wife was going in to deliver our second child. We were talking with the anesthesiologist, and so we got the anesthesiologist resident, who's going to be talking in there too. We talked to the attending later on, but we can be kind of picky at times. But really nice guy, really jovial, good bedside manner. And then my wife said, "Well, what anesthetic are you going to use?" And then when he gave the name, she's like, "So what's the half-life?" Just asked this detailed pharmacokinetics question, and he changed. It's just like, it was like he was back in class again, and you could see that because he was kind of fresh out of med school. Yeah. And it was like, yeah. So we were joking with the attending. Now he was really good. He knew the answers. He was on top of everything. But and we were of course friendly and joking about it. But it's like, I guess we could be like Karen and Mr. Karen or something now. But there's a fine line there. So having that knowledge and kind of figuring it out becomes important. The one other thing I want to talk about too before I let you guys go is a little bit about the FDA. So I used to joke when I played all this basketball that I was like this close to being able to palm the ball. If you had like the right humidity and temperature, I could palm the ball, and that was all perfect. But if it got a little bit outside that area too, or the ball was older or something like that, I just couldn't quite do it. So I used to joke I had thalidomide fingers. So thalidomide was I forget what's prescribed for, but it's
prescribed for like nausea. And
yeah. So a lot of pregnant women would be given thalidomide, particularly in Europe. But there was one woman in the FDA who looked at some data and said, "I don't think this is like held up the approval process in the US. So not very many people in the US were prescribed thalidomide. Meanwhile, in Europe, it was all over the place, and the children, when they're born, like flipper babies, because their limbs just didn't develop. So that was actually a case of one person at the FDA actually using their authority, and even though they were under pressure to try and approve this, saying, no, we need more study on this, and it essentially made the thalidomide situation in the US far, far, far better than it could have been. It could have been much, much worse. But anyway, so again, I have a science bias. You got that logical thinking, and you can look at stuff and figure it out, and advocate for yourself as well too. And this kind of becomes a responsibility. And it's like my poor wife sometimes has friends and relatives call her up and say, hey, I got prescribed this drug, what can you tell me about? But she will do that. And then I know sometimes in pharmacy she's done volunteer work where bring in your drugs and let's take a look at what all you're taking, particularly for like older people in various like churches. You got like some old person who's got like a dozen prescriptions. It's like, well, let's take a look at this and see if this is appropriate. So it can be hard to keep track of that. And how does that relate to this? Well, there's a lot of stuff to keep track of here, especially when we get to chapter 23 and the combined, holistic regulation, combined regulation. It's crazy. What happens if you tweak this? But on the bright side, this is why there's definitely a take-home version of the exam coming up. I have a question that I love so much. It's like, this is the best question ever. And now I'm figuring out how to add to it. And it doesn't have a right answer. And I just want to see what you guys come up with for it. So you can't get it wrong. But yeah, so it's ridiculous too. But anyway, that's all I wanted to do today. So